Welcome to a special episode, a COVID-19 pandemic episode of The Future of Everything. I'm Russ Altman, and I would like to say to start out with that we are taping in non-professional conditions, so I want to apologize in advance for any non-optimal audio, but still we're very excited to be here, and we're excited to be talking today with Dr. Catherine Blish. She's a professor of medicine and is expert in infectious disease and in immunology at Stanford University. We are speaking late at night because Dr. Blish has been working extremely hard in response to this pandemic, and that's the topic for today. So, Catherine, thanks very much for being with us in the middle of the night.
No problem. It's my pleasure to be here. Thank you, Russ.
So I wanted to start out just to get a sense of what is your normal work as a professor at Stanford, and how has it changed in the last couple of months because of the onset of this pandemic?
Well, that's a really good question. My normal work is that I run a research lab that is focused on the immunology of infectious diseases, and specifically we're really interested in studying how different infections interact with our body's host defenses. We normally work on a range of pathogens. Right now, the pathogens of the day had been, prior to this, HIV, tuberculosis, dengue virus, influenza, and Zika. So we have a pretty broad-ranging array of pathogens.
Yes.
So I split my time between running that research lab. I also co-direct our MD PhD program, so I work a lot with our students. I do some teaching in our graduate level immunology courses, and I do some clinical work in infectious diseases. So that's my normal life.
Yes, and so I take it something happened and something changed.
Indeed. And in some regards, it's perhaps less of a change for me than for everyone else in the world, in that we have completely altered our research program, in part because of the shutdown. We closed all our other research programs, and we're entirely focused on trying to understand the pathogenesis of the SARS-CoV-2 virus that causes COVID-19.
And in fact, my understanding is that the labs at Stanford Medical School, unless they were working on COVID, they all had to shut down, put everything in the freezer, or do whatever was necessary. And so it was kind of lucky that your group was in a position where you could keep it going.
Absolutely. And it wasn't entirely seamless. When the shelter in place came down, we still had ongoing tuberculosis experiments where we had to, like, that day kill month-long cultures. But at the same time, we were already starting to build a COVID program from really when we first started hearing about this in January. We thought we'd start building some tools. And we were in a unique position with our tuberculosis program in that we already were familiar with working with what's referred to as a respiratory BSL-3 containment, meaning...
Yeah, so I did want to ask you about that because this is something that I don't know anything about, but there's these three letters, BSL, and then there's 1, 2, 3 there. So could you just quickly give those of us who are not familiar with this nomenclature, what are we talking about and why is this important?
Absolutely. So BSL stands for biosafety level.
Okay, good.
And so BSL-1 means the lowest level of biosafety level. So BSL-1 is anything that involves some risk of potential transmissible infection, but a very low risk. So for instance, any work with human blood or blood products would be considered a BSL-1.
Because you don't know what might be in the blood.
Yes. And in fact, we're supposed to always use what are referred to as universal precautions with blood or any other body fluid because you don't know what anyone has. BSL-2 is the next level up. That would be a virus like most of those we work with normally, HIV, influenza, dengue virus, where we know that this could be transmissible, but it's not likely to happen based on just working with this in the lab. So for BSL-2, you always have to do your work in what are referred to as these biological safety cabinets, which are basically these big metal canisters that have an airflow going through them, so that in theory, you're not breathing what you're working with.
Because they'll be like sucking air, kind of like what, Similar to what happens at toll booths, but maybe in an opposite direction. But I know that they always have to protect the toll booth people from the exhaust, and you have to protect your workers in your lab from inhaling the bugs that they're working with.
Exactly. And the way it works with these BSL cabinets is that it protects the worker, and then they all go through a bunch of filters before any of that air gets exhausted.
Right, and that's good for those of us in the general population to know.
Exactly. Now, BSL...
So this, I just want to say, BSL-2, those are some serious viruses that you mentioned just now. So BSL-2 already is clearly a non-trivial situation that requires some care. But my understanding is that that is not what is needed for COVID and friends.
That's correct. And in fact, so BSL-3 is the next level up, and that is a whole huge step in the containment. And in fact, large quantities, like if I decided to grow up, 20 gallons of HIV, that would not be BSL-2. That suddenly becomes BSL-3. Like you're not supposed to have huge quantities. But BSL-3 means that not only do you need these biologic safety cabinets, but you need to be protected. So in this case, and then there's actually levels within that. There's respiratory BSL-3, which is what COVID is, which basically means that this is an airborne pathogen. It can travel through the air, and everyone who works in it basically is wearing a spacesuit.
Okay.
So they're head to toe covered in Tyvek or plastic. They've got a respirator that is constantly recirculating air with a big mask in front of them. They're wearing several sets of gloves, and they're still doing all their work inside one of those cabinets.
Wow.
And
okay.
It has to be in a negative pressure room, meaning that no air can ever escape that room without being filtered a gazillion times over.
And this whole, thank you very much. So that whole explanation is because you mentioned these letters and you said BSL-3. So presumably the work that's going on now, did Stanford have plenty of these BSL-3 facilities that you could perhaps that you were already using or that you could commandeer? What was the situation? It sounds like they're probably expensive, for example.
They're incredibly expensive. And Stanford, relative to our peer institutions, is very poorly equipped in BSL-3 space for a whole variety of reasons, part of which being that we happen to live in one of the most expensive areas in the country and their expensive facilities, and part of it historical and so forth. But we had a small BSL-3 facility that Carolyn Bertozzi, myself, and a few others had been using for work with tuberculosis, which is another BSL-3 pathogen. And in the months or so that COVID was looking like a serious pathogen, I had been in conversation with Carolyn and others about the possibility of converting that into a COVID facility instead of a tuberculosis facility. Up until this point, there's generally a theory that you don't want to mix bacterial and viral pathogens.
Right. You could see that because of contamination issues and...
Yeah, and there are some details about disinfectants you use and all sorts of things.
Right.
But we were in the process of trying to figure out whether to make a full transition or to introduce both at once, which was problematic when the shelter in place announced, at which point basically we killed everything TB, and Carolyn handed the facility over to me to turn it into a SARS-CoV-2 facility for COVID.
Great. Okay, so now you have the space to work. Tell us what are and this is what I'm sure everybody's wondering, what are the questions that you're trying to address? What are we learning with this facility and with your capabilities?
So a lot of things. There's so much we don't understand about this virus. There's a lot we can infer from the original SARS, which is now called SARS-CoV-1, but a lot we don't know. So we're starting with very simple things. Which cells does it infect? How does it infect them? And what happens to those cells when they become infected?
So even that is not perfectly clear yet.
It's absolutely not perfectly clear. And we have a lot of really fabulous scientists at Stanford who have done things like build these lung organoids or little mini lungs. And so we'll be doing things like infecting lung organoids, infecting gut organoids, because there's a lot of debate whether this virus actually infects the gut as well as other human lung samples and other tissues. There's interest in why these smell and taste issues, is it infecting olfactory or smell cells? So a lot of it is important to understand the basics of the virus. The other issue is to build the most physiologic, meaning the best mimicking of the natural infection system, where we can begin to do drug screens.
Mm-hmm. Of great importance, obviously.
Yeah, and in particular there, in terms of acting quickly, it's great to find totally new mechanisms and totally new drugs, and that's a lot of what my lab normally does. But when we're talking pandemic, we want something that we can get into people very quickly. So for this, what we're looking at a lot is repurposed drugs that have been evaluated for other things that we know are safe to give to people, and to see if they can inhibit the virus, but not just in tissue culture, in a more physiologic system.
So when you say tissue culture, you mean like basically, forgive my simplicity, but a Petri dish with cells growing, you add something and it protects those cells from the virus. That would build some confidence, but might not be enough for a doctor to say, I'm going to give this to my patient.
Precisely. And that can be for a whole variety of reasons. And I think this whole hydroxychloroquine story is the perfect example of this, because honestly, I don't know that I can name a virus that hydroxychloroquine doesn't inhibit its replication in tissue culture, meaning on these boring little cells that are laying flat on a dish. And that's partly because the drug is somewhat toxic to the cells. And if the cells don't grow well, the virus doesn't grow well.
So it weakens the cells, and then the virus can't do as well in a broken cell.
Yeah.
Because viruses often commandeer the cellular machinery to do their thing.
Exactly. And that can work really well in a person, but it may not. And the only way to really know that is to either have a better system to test it ahead of time, or to go ahead and test it in people, which is what's happening now. But what we'd really like to do is build better systems so we don't, say, cause fatal arrhythmias in testing it in people, but figure out that maybe it's not the best option right now.
So are those tests already going? Or I would guess there's a lot of setup required. So where are we in having a system that you would trust to say, okay, if I give a test medication to this system, and if it works, I'm pretty excited, and I'm going to call up my clinical trial colleagues to start gearing up. And we'll get to clinical trials, I'm sure, later. But so where are we in that process, you and also the world?
Well, we are really, really early, and nowhere close. I will be perfectly honest. This is a virus that I had personally never worked with before, nor had really anyone else. Although at least the groups like Ralph Barrick, who's a world expert on coronaviruses, they were at least a little more familiar with it. We're still growing the virus and titering it. And so we need to test a bunch of systems in order to do it. And each, just to sort of give an idea of perspective, it takes 3-4 days to grow the virus, another 3 to 4 days to figure out if what you grew is any good. And then 3 to 4 days to evaluate everything you sort of chuck that virus on to see if it works.
So this may seem impertinent, but where are you actually getting the virus from? And is that a challenge or not? Is it all over the place? Are there plentiful supplies, or is this an actual logistical challenge?
We got in early enough that we were okay. So the National Institutes of Health, and specifically NIAID, the National Institute of Allergy and Infectious Diseases.
That's the one led by the famous Tony Fauci.
By the famous Tony Fauci.
Okay, just want to make sure we're all on board with that.
Yes. So they have basically almost a company-like thing that they support that exclusively exists to share viruses and reagents among researchers. And early on, we requested a vial of SARS-CoV-2 from them. And the strain we have was from the first Seattle patient
Wow.
that they grew and have distributed around the country. And, it wasn't particularly high titer, we did discover, so we're still trying to grow up more virus so we have enough to do like big experiments. But that's the strain a lot of people are using. You can also try to grow it out of people. That gets more complicated because you have to worry about other viruses also growing.
Yes, so there's a purity issue. Yeah, I'm a little bit surprised because I thought you were going to say that since Santa Clara County and Stanford was one of the early hotspots, that you had grabbed it from all the patients coming to our hospital, but that was not that was
No.
not the case.
I mean, we could have done that, I suppose, but it was easier just to put in an online order for BEI and get the vial from the even earlier Seattle patient.
Now, is there any worry about evolution and change of the virus over time? So the worry would be that whatever showed up in Seattle a couple of months ago may be different from what other doctors are dealing with in New York or in Louisiana or Santa Clara County.
Absolutely. And one thing I should mention is in conjunction with all the pathogenesis work, we're also running a huge biobanking effort in which we are collecting samples from all the COVID patients seen at Stanford Hospital and hopefully expanding out in the community, but we'll see. For more than a month now. And
great. So that will give you that data about how it may have evolved over time.
Absolutely. And we're already getting results from those studies in which, because it's also nice to be able to, we can build all these systems in our little dishes, but it's nice to be able to go back and forth. And this is something that my lab has always done is we try to build these little model systems in dishes. But ultimately, you need ground truth. And to do that, we tend to recruit cohorts of the people and make sure that what we're seeing in a dish actually resembles what we're seeing in a person.
In terms of the infectivity process and the cells that are changing and the cells that are responding to the infection.
Yeah. So there was an interesting preprint a couple days ago showing some evolution of the virus across patients. And we will have similar data within our Stanford cohort soon. It wasn't huge. And it's also important that unlike a lot of viruses, RNA viruses in particular that we think of as really changing very quickly, like HIV, flu.
Yes.
Coronaviruses are massive. They have this huge genome, lots of nucleotides. And as a result, they can't screw up themselves too much. So one of the genes that it encodes is a proofreading gene.
Proofreading, to make sure it's getting all of the DNA right or the RNA.
It literally is scanning to make sure it's not making mistakes. Now, all RNA viruses evolve.
Yes.
Period. But it may evolve a little less quickly than, say, influenza because it has this proofreading.
And that would be, obviously, my sense is very good news in terms of not escaping our medical treatments or escaping a potentially novel vaccine.
Absolutely.
Well, this is the future of everything. I'm Russ Altman, more with Dr. Catherine Blish about the battle against COVID-19 and the SARS-CoV-2 virus next on Sirius XM. Welcome back to the future of everything. I'm Russ Altman, and I'm speaking with Dr. Catherine Blish from Stanford University, an expert in both immunology and infectious disease, whose life has been rocked with the COVID, as many of us lives have, by the COVID-19 pandemic. So you began to tell us some intriguing things about what's beginning to be learned in your facilities, but I wanted to ask, as somebody who's both a physician and a researcher, what should we know about this virus? Is this virus looking like the other viruses you've studied? You actually, at the beginning of our conversation, listed several famous viruses ranging from Zika to HIV and others. Where does this fall? Is it matching your intuitions about how viruses work? And where are the surprises so far?
So it's a really great question. And in terms of, I like the way you put that, matching my intuition about how viruses work. is an intriguing way because I spend a lot of time thinking about how viruses work and often make the analogy that our viruses know us better than we know ourselves because they have all evolved mechanisms to try to escape from our immune response. And so they all do that. And that is the commonality, and that is what makes us a great virus. But what's fascinating is the different paths by which each virus reaches that point.
Yes, yes.
And,
different strokes for different viruses.
yeah. And what we've seen in the early data has been interesting in that the SARS-CoV-2 virus that causes COVID has taken some unique paths to its interaction with our immune system. And the first thing to know is that we profiled the immune response of seven people in 8 samples who all had what we classify as severe disease. Right. Meaning they required hospitalization, but these were actually on the border of more severe. Most were in the intensive care unit, and half of the samples were drawn when people were on ventilators, requiring breathing machines, and in what we refer to as acute respiratory distress syndrome.
Yeah.
And we saw a complete and massive reshaping of the immune response in all 8 of these patients, all 8 of these subjects, rather.
Yes.
But particularly dramatic in those who were all the way into what we refer to as ARDS. But what was really interesting was seeing that the virus has taken what I think will turn out to be a dramatic escape mechanism. Oh. In that there's a molecule on many of our immune cells that called the major histocompatibility antigen, MHC, and there are a couple flavors of this, well, actually lots of flavors, but half of it, basically, their job is to talk to T cells, another important component of our immune system, and absolutely vital for vaccines and memory. And basically what MHC does is it tells the T cells, here's a piece of this virus that you should respond to.
Yeah,
so the more severe the infection was, the less of the MHC you saw. So,
which is the opposite of what should be happening?
Absolutely the opposite. So what that's doing is it's probably inhibiting the really important role of particularly the CD4 T cells, which people might have heard of from HIV. They're the orchestrator of the immune response, and they're the cell that HIV decimates, which tells you how important they are. But in the setting of severe COVID, those cells are probably not receiving the instruction they need. Because
so that's a big one. And is this the first time you've seen this kind of response from a virus infection, or has this been seen before?
So interestingly enough, when we saw this, we looked back, and though it looks like this is a common trait of coronaviruses, which I haven't worked with before, but the original SARS and MERS both did this as well. So it makes sense, but this is the first time we've seen it with the SARS-CoV-2. The second fascinating thing that we've seen is that there's been a lot of attention, you might have heard this term, cytokine storm.
Yep.
Where there's this concern that it's really a poorly regulated immune response that's secreting all this junk that's causing too much inflammation that's actually making people sick. And that probably is an important part of how this virus is causing disease and severe infection. But most of my colleagues in critical care are a little less convinced than the people who aren't in critical care, because while this is happening, the levels aren't quite as high as we sometimes see in other cases of this ARDS. So I think there's a more nuanced story there. And consistent with that, we expected that these blood cells would be secreting tons of these pro-inflammatory cytokines if there was a cytokine storm. And really, we didn't observe that at all. There was almost nothing. So if there is cytokine storm, it's happening locally in the lung, and we're not seeing it in the peripherally circulating cells.
I see. So the immunological mayhem looks different from the mayhem from other viruses and other diseases of the lungs. Oh, this is really interesting stuff. Are there any more surprises?
Well, so there are arguably two more. One is there have been reports that the T cells, those coordinating cells, are so-called exhausted. And that's potentially related to the cytokine storm. We didn't see a ton of that, but we saw that another cell called a natural killer cell, which is related to them, but maybe a faster activation.
And which I happen to know you're a world expert on.
Perhaps. I love my NK cells. So the problem is that there aren't many NK cells to study in COVID because they're mostly gone. They're decimated. But the ones that are there do look exhausted. Okay,
so there's an exhausted natural killer, and that seems to be bad.
We think so. Although, and interestingly enough, there are some studies out of China showing that there is NK cell infiltration in the lungs. So the reason they're gone from the blood may be that they're where the action is.
Okay.
Which may be the same with the T cells. And then the final thing that we saw that was completely weird is that in the most severe cases of infection, it appeared that the B cells, which normally we know make antibodies.
Yep, these are the antibodies, the famous antibody-making cells.
Yep, it appears that a subset of them decided to change their mind about what they were going to be when they grew up. And they turned into a type of cell referred to as a granulocyte, basically a neutrophil, these fast-acting innate immune cells that are in a completely different arm of the immune response than B cells. And this isn't technically supposed to happen, but there have been other reports. But
so this was like a fate switch. They were going down a path. They were supposed to turn into cells that made antibodies, which are normally good because the antibodies then bind the cells that are infected and get them out of the system. And it's, if I'm understanding you, you're suggesting that maybe the virus has figured out how to get them to go down another path that is not nearly as dangerous to the virus ultimately. I'm extrapolating from what you just said.
Yeah, I mean, maybe slightly more teleological. Yeah. You know, I don't know if the virus did it on purpose. And to be fair, these B cells had already become B cells. They were already making antibodies, but then they decided to become granulocytes, which is completely weird.
So as we finish up, thank you. So you just gave us 4 tantalizing and confusing, but it's the reality of where you are right now, which is really more than we could ask. So, but just to finish up, give us the outlook about what is the next several weeks to months look like for discovery scientists like yourself who are just trying to get down to the bottom of this virus. So I know you can't read a crystal ball, but how are you imagining it might go? And we'll look back in months and we'll laugh and we'll say, well, that's what we thought. But in closing, can you give us a sense of where we're headed?
Absolutely. So science, as much as we would love it, doesn't always act as quickly as we would like, but I would love to think that there's been a really lovely group mentality about this. And I think in a few months, we will have additional drug candidates that we've identified through screens that will be going into clinical trials and may have an impact. The other thing we will have is results from the first human vaccine trials that started, and really, ultimately, a vaccine is going to be the best way to get out of this.
Yes.
So that's the optimistic view. And I do want to give a shout out to my colleagues in the Bay Area. We formed a BSL-3 consortium with Berkeley, UCSF, Davis, Gladstone. Everyone's sharing reagents, equipment, which is like gold these days.
Yeah, and it's been a theme going through a lot of our conversations is that people have come together who normally might be competitive or not even interact with one another. And there's this great sense of like, let's do this together, which is quite heartening for those of us who are watching.
Absolutely. It's been just fantastic.
Well, thank you for listening to The Future of Everything. I'm Russ Altman. If you missed any of this episode, listen anytime on demand with the SiriusXM app. Stay safe, stay healthy, everybody.